Well, welcome back, everyone, to the industry panel discussion. So we're going to start by, well, the panel are going to introduce themselves. I'm Ashley Norris from Moe Genetics. After the introduction of the panel, I'm going to speak briefly about Moe Genetics and how we use genomics currently and what our wish list is from this project. And can I just start by saying congratulations as well to the project partners on producing a fantastic resource for the industry. Okay, so we have our panel together. So if I can ask the panel to start introducing themselves. Ross?
Yes, hi everyone, Ross Houston. My title is Director of Genetics and Innovation, and I work at Benchmark Genetics, which is an aquaculture breeding and biotechnology company.
Yes, I'm Tim Knudsen. You saw me not long ago, so I'm working in Aqua again as a bioinformatician, and main responsibility there is doing GEVAs, updating, creating SNP arrays for the 4 species that we have breeding programs for. The biggest being salmon and then rainbow trout. Yes, thanks.
Okay, so I'm Dr. Emily Clark. I'm based at the Roslin Institute in Edinburgh, where I'm a research group leader. So I'm a member of the BOVREG project and also co-coordinate the AQUA-FAANG research infrastructure. So I think my job on this panel is to represent larger terrestrial farmed animals.
This is Sara Fajon from the University of Padova. I'm a researcher. I work mainly on selective breeding and genomic selection in aquaculture species.
I'm Antti Kause from Luke, Finland. It's a governmental research institute. We run national breeding programs to help our industry, and I'm also the coordinator of the AQUA-Impact European Union project.
Thank you. Good afternoon. I'm Ian Johnston. I'm the CEO and one of the founders of Select, and my colleagues have done a very good job of giving you an idea of what the company does, so I'll stop there.
Okay, thank you, panel. Okay, so a brief introduction to Moe, and maybe this will frame, what I was going to say a typical aquaculture breeding, but a very large aquaculture breeding would expect from a project like this, or would hope. So as I said, my name is Ashley Norris, and I'm responsible for R&D in Moe Genetics, which is a global group that supports all the Moe breeding programs. But I also have a special responsibility for Ireland and Scotland breeding programs as well. So we're a large company, produce a lot of salmon, only salmon, although we do produce wrasse as a cleaner fish. We do breed wrasse as well. We're 20% of the market, and we produce 8 million salmon meals a day. So I think that, when you think of it like that, it puts it into perspective. Those regions in orange are all the countries that we're represented in. So we're a fully integrated company, and I work in this area. But we're also in all aspects of salmon farming, from feed all the way down to consumer products and marketing. So I think those of us in breeding have a very good idea what the customer wants, because the customer are the people we're sitting beside having lunch with. And our farmers are also very, very well connected to the marketplace, because they're getting feedback directly from our marketeers back to farm level. And we have, Moe Genetics is a group within Moe that supports the 5 breeding programs. So we have breeding programs in Western Canada, Eastern Canada, in Chile and South America, and Norway, of course, and then a kind of joint program between Ireland and Scotland. And this has been shown before here today, which I was glad to see, the breeder's equation. It keeps us all focused on what we can do, because this is our business of Moe Genetics. It's genetic gain, making genetic gain. And we can only do it in 2 ways. We can increase these on top, or we can decrease what's on the bottom. It's as simple as that. So where a project like this comes in, it's, of course, how accurately can we select fish? Or how much more accurately can we select fish compared to what we currently do? So what we do on the farm, because I want to bring it back there, because that's where I spend a lot of time, it's quite simple, really, in terms of genomics. This is the most important thing we do. And those two guys are doing the same thing. They're both, very sorry to the panel having to turn around. They're both taking a DNA sample. And we take probably over 100,000 of these a year throughout Mowi genetics, or throughout Mowi breeding farms. And this guy is taking it from a large fish, so you can't use a machine to do that because the fish are alive and big. And this guy is using a machine we developed to take a DNA sample and input a PID tag. Of course, the fish needs a tag after all of this. So this is what the farm generates when they're involved with the breeding program. For every fish we measure, we take a DNA sample that ends up in a barcoded tube. And that goes to the lab. But that's not what the farm sees. The farm doesn't see the lab. The farm sees just what we deliver back to them. So it's a very smooth process, and it integrates very well with our breeding programs. This is literally the only extra work that the farm people have to do. Of course, there's recording phenotypes, but we mostly do that as a team in Mowi Genetics. Go to farms, or go to processing plants and collect weights, for example, or send fish to challenge tests. And it's very important that a technology like this works in remote locations. It works in very difficult environments as well. They're wet and they're cold. And what we get back from our sample is a genetic link to the parents and to all the other fish, both of this generation and of the previous generation. And actually, Mowi Ireland were the first company in aquaculture, and maybe somebody here could correct me. I'm pretty sure we were the first to ever use genotypes to assign fish to parents. So genotypes in place of tanks, individual tanks. And that was we started that back in 1998. And that was done from pure necessity, which is the mother of invention, literally, because we didn't have the planning permission to build a tank-based facility for 200 tanks in Ireland. And if we had waited then, we still wouldn't have it now. So we started taking DNA samples and running microsatellite assays. I'm not sure if you call that genomics. It's like pre-genomics almost. But that gave us what we needed back then, before the 21st century. It gave us a genetic link between fish. And that enabled us to start a breeding program. And the following year, Norway started the same type of breeding program. This DNA sample also gives us QTL information for some traits, for quite a few traits, actually. And that's by far the preferred selection source, if you like, especially when it comes down to people on the farms, because you can very easily divide up fish into 3 groups. Breeding values are a little bit more complex, but obviously not all traits have significant QTLs. And finally, what we get from the sample is a genomic breeding value from a GWAS based on a 55,000 SNP array. So we get all that back from that one sample. And what we do with that, for our 5 breeding programs, we provide different kinds of information depending on the trait they're interested in and depending on what facilities they have to divide up different fish, and et cetera. There's a number of different factors here. But you can see that it's a mixture of genomic breeding values and QTLs for different regions. With the exception of Canada East, they still use individual family tanks, and they use family estimated breeding values. You might also notice from here a lot of the traits, the majority of the traits are disease resistance. And even though most of the breeding programs started with growth and maybe late maturation, now we are adding more and more disease resistance traits because that's the nature of our business, unfortunately. So most of these disease resistance traits are also based on challenge tests. And that's, as a number of people have mentioned, that's becoming a larger and larger difficulty for a company. There's the cost element, and not just the costs of the challenge test, but when you run a challenge test, you're not just using QTL, so you have to do the whole array. You have to genotype for the entire genome. But we're facing more and more restrictions doing challenge tests every year from the authorities. They also don't always work. I never heard anybody else say that, but maybe we're the only ones who don't always get a challenge test to work effectively. We have one that has stopped working. Now, we think it's because we selected the fish so well over so many years that they simply are pure resistant. But I suspect it's actually a couple of factors at play. So if you were to ask us what one of the future challenges for our breeding programs are, it's not the price of the genotyping. That has come down massively, especially as Ian said, it's not the number of SNPs, it's the number of fish. But one of our biggest worries going forward is, will we be able to run challenge tests? Or will we be able to run them the size that we currently run them? So I just asked a few people few people from our company at the Fish Breeders Roundtable that was on this week in Bergen. What do you want? First of all, just better genomics. We have genomics, it works really well, it's really efficient. We can have thousands of fish sampled a day. We can have those samples in the lab the next day, and we can have the results in 2 weeks. It's just really, really works well. But we want it to be better. We want better markers. We want them to explain more of the variation. We would like to do more marker-assisted selection. So bigger and better. We'd like to understand gene pathways and functions so that we can optimize future, well, for example, gene edits. Everyone's talking about gene editing, and it's very possible, and it's out there. But we don't really know what to edit other than the obvious ones like sterility. There's a very complex pathway to sea lice resistance, and we have a number of projects that are working on that now and looking at different species, but how will that integrate with AQUA-FAANG results, for example? Disease phenotypes without challenge tests, is that too much to ask? You know, I mean, who's looking at challenge tests other than us just taking hearts out to examine for histopathology? The rest of the fish is getting thrown away. There must be some more we can do there. You know, it's such an incredible resource. And finally, how do our data people, those people integrate functional genomics into our genomic prediction models? Now, I'm asking that because I don't understand how they'll do it, but I imagine they probably do understand how they will do it. But I was thinking, you know, what is the communication flow? What is the availability of this resource going to be like for people working in industry? So there are all the questions I have. So I'm going to put a couple of questions to us all as a group, and feel free to answer or ask more questions. The panel can answer, I can answer. So the first question is, what tools will this project deliver in the short to medium term for the industry? And I'm thinking, like, will we be doing something different in 1 year or 2 years' time? And the industry needs to know, because we design the arrays and then we buy a whole load of them. So there's going to be a lag between knowing what to change in your SNP arrays from now until when new ones can be designed.
Yeah, just because I was involved in Work Package 5, which is probably the most related to industry, work package of AQUA-FAANG. And I think that the detection of the major QTL could be a very good start in the short term. So we are going forward this direction with the designing of the SNP chip. So this could be really important for the short term and for the application in very few months directly into aquaculture farms. So everybody can buy this SNP chip, well, not buy, but genotype using this SNP chip, and they can have their resistant animals in the short term. Well, not in the short term, but I mean, they can start using this tool to select their animals. So I think this could be really the first results that could be really applicable to the industry.
But is the annotation done by, for example, by trait, by disease, or do you first need to have a starting point?
What do you mean?
Like, for example, we have a lot of information about,
Yeah.
CMS, the disease CMS, and we know
Yes.
what QTLs are involved, and we know what chromosomes are on.
Yes.
So you need that information as a starting point, and then you go to that region of the annotated genome to
Yes.
find what closer markers you could use.
Well, the QTL has been already detected, so we can really use this information that has been, obtained during AQUA-FAANG. So I think it could be really a good start, and it could be the start to design this SNP chip. So, this is in the very short term.
Yeah, okay. Anyone else have a comment about that? Oh, I should pass around.
Yeah, just to comment, in Work Package 3, we deliberately designed agnostic immunological stimulations to bacterial and virus. So effectively, this is identifying genome regions that are being activated by a viral stimulant. And the rationale was that should transfer across to multiple different viral pathogens. Noting every host pathogen interaction will have its own nuances, but these regions should have general applicability. So I would imagine if one was to prioritize SNPs hitting those regions across the genome, I'm not quite sure what the direction of effect will be, but they will have an impact more strongly by a long way than random SNPs would. So a SNP array focused on those would be, I think, very interesting to test the prediction accuracy, particularly across more distantly related animals for viral diseases. I think that Tim is going to effectively try that as a test, and it'd be interesting to see more broadly.
Just say for immediate impact, I wouldn't underestimate the benefits that will come from just having better quality genomes for those 6 species, because that's kind of foundational for all tool development, and that will be really quick and have a really big impact, even before we get to the heady heights of good functional annotation of those genomes.
Do you want to pass that down? Sorry, Tim was first.
Okay, so this is following up on what you said. So it's great if these general disease-resistant regions will end up working for several viral and bacterial traits. But then you kind of, if that can cover the diseases, that's great. But then again, most important is maybe growth. So then the question is, will there be any overlap between these regulatory regions that you find for disease or maturation with the very polygenic but very important trait like growth? And do you have any data that can show that? if the degree of overlap between like the different data sets that you have in different tissues, and like, does anyone know?
So yes, we can absolutely say exactly where the activity of these elements are across different tissues. So we can't necessarily link that to growth, but if you take a sensible approach and look at liver or muscle or tissues that are involved in growth, so that can be done. My question that I have more generally when I think about a low density panel, but the need to select for multiple traits in a breeding program, is how do we prioritize SNPs to target multiple traits based on these regions? It sounds more plausible to do that for particular areas, but that's not how industry is working. And then it seems like you'd need to increase the density of markers further, which seems like diminishing returns based on how things is working with the actual breeding program. So I think that's an interesting challenge.
Yeah. Sig has a question. Gary Ross.
No, it was just to answer your question. So I think I agree that just the improvement of the annotated genomes and the availability of the resources and the databases are just fundamentally useful for all of us. So that's academia and industry. Maybe some of it's in the short term. Some of it we know about in the long term, particularly gene editing and understanding the biology of gene editing targets and finding those targets. But maybe some of it we don't know yet, because we don't know what new technology is going to come along. But we do know that improved knowledge of the genomes of these species and how they're regulated are fundamentally important. My comment is on the other comment is on the disease challenge testing. So from what I can see, and not just here in this project, but also from the literature, when you're predicting across populations that are more distant than, say, half-siblings, the prediction accuracy is very poor unless you have a major QTL, in which case you can get good prediction accuracy. And I think that's partly why you're getting good prediction accuracy, is because it's big QTLs. And then if you combine that with your breeder's equation, which we like to see, and also the number of traits that you're focusing on, right? So you have to put, let's say for argument's sake, 50% of your breeding goal on growth. And so all that's left is a few percent for some of these disease traits. Some of them you can maybe only put 10% of your breeding goal on.
In the nucleus, you're speaking.
Perhaps in the nucleus, it could be more intense in the multiplication line, sure. And then if you have your accuracy dropping down to levels that are far below what we're having with the SIB selection, which let's say on average is about 0.7, but if you take that down and you can only get 0.2, 0.3, then I don't believe, and we don't know the correlation between the challenge test and the field is 1. It's probably not 1. It's probably also 0.5 or something. You start doing those multiplications, I just don't believe that there is a case for using genomic prediction at the moment to skip a generation of challenge testing.
Except with very large QTLs.
In the case of very large QTLs, and in defense of microsatellites, the IPN QTL was found by microsatellites, and the marker-assisted selection was done with microsatellites.
Yeah, so one quite recent paper in cattle from Mike Goddard's group, I would like to mention that because the approach they are doing, that's a paper published in Nature Communication in 2021, I believe, Chiang. That's a very interesting paper. What they do there, they do very high-powered GWAS studies. For some of the industries here, they have those data already do it. So do it across populations and generations to find SNPs associated with multiple traits and a combination of different traits. And they combine that with information about conserved regions and also regulatory regions. And doing that combination to select the SNPs that are likely functional, maybe causal, they develop arrays that are do a very good prediction for milk production traits, even pleiotropic effects
In all populations.
across populations and breeds in Australia, Europe, and US. So there is a lot of development of new tools to use this data. But I think you need a combination of GWAS and functional annotation and also conserved regions. And I think thinking about diseases and all the diseases we have in aquaculture, from what I have seen from some of the QTL findings, I think you find some of these QTLs in the same regions for multiple diseases, don't you?
Not off the top of my head. Well, the major QTL for diseases in salmon is CMS. It's not the same as IPN. It's one chromosome different.
No, but
27, 26, 27.
But I think these data, the functional or the regulatory regions, it's not like targeting the main QTLs. It's not the most important. If you do this very high-powered GWAS, you see that there is a lot of other SNPs associated with these traits for maturation or for other diseases. And for those, they can be very effective for predicting across populations. At least that study in cattle is kind of promising to do that.
And they have that cooperation across the different companies.
Yes, so they produce data across breeds because people collaborate, you know. So they get data from different populations and different countries.
Is that likely to happen in aquaculture? But that's a question for another day, perhaps. Emily?
Yeah, I could just add about the cattle community, I guess. So most of that work from Australia came from what was called the 1,000 Bulls project, where it was a concept where if you generated a lot of sequence data, you could buy into the consortium and then be part of a really large amount of analysis. And that consortium is now, or the 1,000 Bulls has reached the end of its sort of funding and term, and it is now difficult, I think, to keep it going. It's become the Bovine Longread Consortium. But it really, it led to a lot of papers. The one Sig mentioned, the two I mentioned on Friday, trying to rank how useful different types of functional information were and using a concept called fate scores for which is the best type of functional information for genomic prediction, and was also a precursor, I guess, for farm GTEx, and cattle GTEx, leveraging gene expression information at scale, looking at which were the most important tissues, which were the most informative bits of functional annotation information, and leveraging a lot of public data. I'd be interested in how that translates into aquaculture, I guess.
Well, there's many species in aquaculture, that's the difference, of course. And within each species, there's many different strains as well.
So in cattle, there's also, I guess, Bos taurus, Bos indicus, and I think it does seem to work across Bos indicus, but there's much more noise. And colleagues are looking at cattle, particularly in the African continent, and how applicable it is there. Similar, not completely different.
When we talk about aquaculture, though, we're including, bivalves. And I think there's greater genetic distance between bivalves and finfish than there is between finfish and ourselves.
Within bivalves, there's enough genetic variation to keep you going for infinity.
Yeah. So sorry. Sig.
Thank you. I wanted to comment on the disease challenge and disease phenotypes because we've seen in AQUA-FAANG that there was some disease-like challenges with viral mimics or bacterial mimics to identify functional regions. I'm not sure we have seen the correlation with the actual disease that the breeders have been facing, and so I think it could be useful to have another step of validation of those functional region in actual condition. But that does mean doing more challenge tests. That, as I agree, is more and more
and more
difficult.
Real sampling as well, because that is something that we do. We go out to see, And we know the genotypes a lot of the time because all the males in the whole company are genotyped now. And we track performance in the sea. And it's very important to be able to demonstrate to the powers that be, in whatever industry you're in, what you're doing in this black box of genetics is actually having an impact.
I think the end goal really must be the phenotype, so the survival of the fish, and not, well, the attack peak would be a way to do that, but not the end goal. I think we are missing a link to the end goal. And I must say that developing new challenge tests without like killing the animals would be very crucial also, because this is something that, as Ross mentioned, we need to do every generation. So that's a lot of challenge tests. That's very costly, and also for animal welfare and ethical reasons, it's getting more and more difficult, for probably good reasons, to do.
So you mean challenge tests in the farm?
I mean any challenge that could improve the survival at the farm. Might it be with viral mimics or bacterial mimics, or directly on the farm, for sure.
Sig again.
Just to mention something different than challenge testing. So in vitro methods for this. So there are no using CRISPR screening to do this. Could be an alternative way. It's not replacing what we do with challenge testing, but it's an alternative way of maybe identifying genes of importance for diseases. So this is not something immediate we can use, but maybe I think really in the future this will be something that will help the industry to be more sustainable and avoid this animal testing, because I think this is going to be a huge problem for the industry.
Yeah, I think it is too.
Yeah, I agree, and I think so my point is a purely pragmatic one. I'm not sure we would actually be worth doing the selection for some of these disease traits. If you start to have accuracies that are low because you're predicting across population, you're actually not making a significant enough difference to the trait to be seen in the field. And so that, I think for some of these traits we're at that level already, but I agree that sort of in vitro testing, there was a study, and I can't remember the full author list, but I know Jorge Del Pozo was involved from Edinburgh, and they were looking at the heart of embryonic fish as a sort of proxy of a PD resistance. And I think that sort of thing's quite interesting, to look at those sort of early life or embryonic models to see if they're correlated with challenge test outcomes on the SIBs of the candidates.
Yeah. Another area of interest I see developing on our farms is the use of better phenotyping in the water of fish, and correlating that to the fish identity. So we know that for most diseases, there are, the animal develops in a particular way, or loses weight, or has very small lesions on them. There are possibilities of detecting those as a substitute phenotype. But anyway, to answer our first question, I think the answer is here and now. Like the tools are here and now. It's just up to us to use them, really. But our second topic we wanted to touch on, and it kind of runs into the third, really, and seeing as we're a bit short of time, I'll ask the two of them together. What are the implications for the longer term use of this technology? What will the implications for aquaculture breeding be? And what do we need to do to get there? What steps do we need to take, or what other projects do we need to have in mind if we want to get to that future? Tim?
So I'm very hands-on with all this data, and it's really one very important thing is to be able to have software that can handle everything in a very efficient manner. So we have now recently moved all our data and analysis over to cloud-based platform, and then you pay per hour. And if incorporating all this means that the computation time would be 10 times, then we have to pay for that as well, right? And then that it becomes a very big challenge. So, and I see all these papers being published on efficient methods to handle and making things faster, but very often they're not providing a software or a method. It's just a bunch of equations. And but the Australian group is very good at also providing software. So
And is this because of the sheer number of?
Yeah, and additional layers of stuff. And if you're doing Bayesian modeling, I did for this test like 30, 40 runs of Markov chain Monte Carlo. Like it takes so long. And what we really want is to use all the data, but it becomes almost impossible if we don't have really good software as well. So
and he has the answer to that.
No, not an answer, but I would want to bring one aspect to this long-term view, is education. And at least around the people that we work, then we typically see a lack of skilled people who have expertise on analyzing these very complex data sets, huge data sets. They need skills in computer sciences and programming, and not just the standard software. I mean, we have, let's say, education for human genetics. They typically use software that you press the buttons and you get the result. What we do is much more complicated, especially when we develop new methods. And it would be nice that in Europe we have education at the universities that really make bioinformatics people and computational people for life sciences. That would be the future generation of researchers and people in the companies. And we compete with, nowadays we compete with companies like Google and Microsoft. The same people that work for them, they could work for us. And it's not an easy situation to be in.
Yes. Ian?
Yes. Excuse me. So maybe there's not an AQUA-FAANG 2.0, but there's a whole series of future AQUA-FAANGs, 2.1, 2.2, etc. And one thing we have touched upon today is the AQUA-FAANG program is really directed towards improving selection accuracy. But even inside that selection accuracy box in the Breeders' equation, as you rightly say, better feeder typing also makes a huge difference. And then it's following through from the next stage to genetic gain, which is really what it's all about. And it's not just genetic gain, it's the value of that genetic gain, to get to Ross's point about the diminishing returns when you have lower heritabilities and a small fraction of your selection index. So there aren't really the decision-making tools and well-worked-out models that enable you to translate it to economic gain. So I think a project like that would be very attractive because you can see the benefit of it. And as far as industry is concerned, if you say, this is the return, then it makes the investment, you can make it on a rational decision to other kinds of capital allocation that you do inside the business.
A project on what specifically? You're talking about,
Sorry?
you're talking about better phenotyping?
Just understanding the whole chain to the value of the economic gain that you're producing. So the things that you do along that chain to produce the best value for the amount of money you spend.
Yep.
To do that as a routine analysis. So if you did that, for example, to take Ross's point, you wouldn't start. I mean, if you had the tools, the decision-making tools, you could do all that on a computer before you started doing any experimental project. And then you could.
Yeah, I think there's been a lot of discussions recently at the fish breeders as well about this the economic value of traits, and anyway, it's a very big subject.
Yeah, so I have another comment actually, or maybe a more thought about how we are doing, or how we want to push the fishes like to produce better, to grow better, to mature earlier, etc., and so on. we want to genotype them and be able to do genome editing on it. I think one of my worry is like, I think it's really important that we know the tip point of all those technology, how far we have or we can push the industry, how far we can push the fishes, and when we are to stop to make sure that there is no collapse at one point of the different population of the different breeding programs, like it did happen quite a lot in cattle. I'm sure Emily can comment on that, because like we see that it did happen in some other species breeding programs. So my question is more like how we can infer that, how we can see where is the tip point and not reach this point and make sure we are still performing good enough in a really long term.
Yeah, so there's a couple of issues there. One is genetic variation. Have we taken away too much genetic variation? My own opinion is that I think aquaculture breeding started late enough compared to other animals. I do think we learned lessons there. And it was started more professionally than maybe the early breeding programs for land animals. Sorry, Emily, I'm not an insult to you or anything. But and also, well, I'm speaking about salmon now. We haven't pushed growth very far. We have pushed it, but nobody wants a larger fish. So we are growing fish in a shorter time. And there is perhaps some associated phenotype from that, as in that are the hearts as healthy as they used to be of the fish? But I don't think we have reached the turkey with the legs collapsing stage, and we don't want to. So that's my optimistic take. Did somebody?
Yeah, so looking into the future, so the long-term needs, I think this is connected to the climate change. So we know that the climate is changing, it's getting warmer. The sea is getting warmer, so it's going to be very, very hot in the water in Ireland and Scotland and Norway as well. And this is producing a new challenge for us, producing aquaculture in this region, because we get more diseases, we get new diseases. Well, the parasites, it's also affecting the way the robustness of the salmon, for example. And we have to move from the sea on land, new production in recirculation systems. I think these are totally new environments and new challenges that we should think about. How can we use genomics to maybe tackle those problems that we know they will come in some years, not immediate, but we see them already coming. Mm-hmm. So I think a FAANG 2.0 is something has to deal with the changing environment and sustainability of the production.
And how will we record that as well? How do we record the stress of animals in these environments, and how the different behaviours in land-based versus in the open sea pens?
And I think that would demand some new set of data, functional data with land-based recirculation. What genes are turned on in those environments versus what you have in sea, for example. I think that's new experiments.
Mm. Yeah, and I presented something at Fish Breeders this week about this. And we were involved in a Scottish study that was looking at the same families in loch and wrasse environments, and we saw very different growth patterns. So I've no doubt that so many different traits are going to be affected. Can you pass the microphone now?
Okay, so one of the ways out of the diminishing return problem with disease resistance is potentially genome editing, because it's done in a different way. So effectively you could take out a disease trait if you're successful in editing for a disease-resistant line. You could take out a disease trait from the breeding gull, for example. Let's say in your sea lice example, the holy grail. Yeah. If you were to able to,
turn
with
it
no
into
other
a coho,
consequences, create a coho-like response in Atlantic, then you don't have to breed for sea lice resistance anymore, so you can put more effort on other traits, and it's a bit of a virtuous cycle. But there's a number of things that need to happen to get there. And I think this is partly where some of the research should go also, is one is to identify the right targets to edit, and I guess that's part of what these projects are doing, but I don't think they're going to get 100% of the way there, so there's going to still be a lot to do. And even when you find a gene, there's a lot to know about how to modify that gene to have the particular phenotype. So editing technology, I think, is, and the intersection of editing technology with that sort of knowledge of regulation of genes, is the other part. You know, it's how to make the editing precise, how to modulate gene expression, how to knock down gene expression, you know, how to be tunable with these sort of things. And I think it's a natural follow-on from some of this work to be able to marry this sort of functional data to a way of validating through gene editing, and at the same time developing the gene editing technologies so they can be, you know, more refined than they currently are. And the other part that needs to happen is the regulatory side, but, you know, we just continue to work away at that.
Yeah, well, we have to continue to keep working at this. And you say there's two parallel, you know, there's one, how is it actually going to be done? How are you going to get edits? And the other one is, what are we going to edit? So they're two pretty big questions, have to be answered.
Two different types of regulatory being discussed. Regulatory elements.
Yes. We're talking about the government type
regulation.
Both are complicated.
So I think my comment follows on from what Ross said, and I think one thing we need to do is acknowledge that AQUA-FAANG's taken a step forward in understanding the regulation of the genome, but it really is just a starting point when we think about the sample diversity that we've captured in the project. We've done some different tissues and we've done these immunological challenges. I think our understanding has taken a step forward, but it's still very far, as Ross says. So we need to start thinking, if we buy into this idea of using functional data, then we're much better at doing it now because of AQUA-FAANG. We can generate this sort of data much more quickly now. We need to think about other traits that we're going after, whether that's certain diseases with different species, so we have which genome regions are important specifically to those. We have lots of blind spots currently, different tissues or stages that we don't know about, and then there's cells where phenotypes actually happen, and the technologies exist. We're better at them now. So this is something to keep in mind from the science side as well, if we buy into this idea, which I think we do.
And keep in mind that you're going to need to collect this data, the actual tissue samples, before the challenge tests are no longer being run on a large scale.
I just wanted to comment on gene editing. I think also a very limiting thing about the technology is the ability to do this very efficient. Because you can't do this once, although that's successful, you have to do it in different genetic backgrounds. You have to do it in many, many individuals. And currently this is very inefficient, the way we do it within the action. So it's not applicable to industry settings as it is today.
Yeah, it's not applicable, but I can see a way that we could, perhaps, that could be. I mean, you know, currently we take eggs and we bath them a number of times with, you know, disinfectant or activators and all sorts, you know, so I do think if you're going to do this large scale, for fish it will need to be bath type, it will need to be something being delivered into the eggs that way. Emily.
Yeah, I could just add, I guess, on the, I think aquaculture has the advantage of scalability in terms of editing. And when you think about terrestrial livestock, their generation times are very long. Doing surgeries on farm is difficult. You have to do laparoscopic surgery for embryonic transfer. Whereas, the potential to move forwards quickly with genome editing in aquaculture seems to be greater, I would say, and potential opportunities to do that. And then, I guess just in terms of scalability and the ability to manipulate environments more easily in more numbers of animals, probably gives significant opportunities in that space, I think.
We have to look at the bright side. Ian.
So, I mean, first of all, you have to have the societal buy-in and get the legal framework. But then the regulatory process is very long. I guess it's going to get quicker with experience, but the first gene-edited farm animal, I think, is the Perth's pig. Sorry?
No, well, gene edited as opposed to GMO.
Gene editing. And I don't know when it started, but I think it's at least 6 years. I mean, somebody from the Rosalind, I think it started in the Rosalind.
At least 8 years ago. At
Eight
least,
years
that's when
ago.
the paper was published, but it could be more.
It's of that order, and the regulatory process is going on in several countries. Apparently 2 weeks ago, Colombia approved it for use in commercial production. The USA might be in the spring, and perhaps China a little bit later. And the main issue is understanding the off-target edits and proving that this animal is safe to let out if it gets out, basically. So it's a long road,
Yes.
but it is going to happen.
I can make a quick comment about the gene editing. I think the progress what we see in plant and crop sciences, that will be the first line of kind of approval for these methods, and they will influence the animal production side, what decisions are made there and what progress is made there. So that's potentially the first step for Europe.
So just to plug the AQUA-FAANG research infrastructure project, so we have a think tank on genome editing, which is a vehicle to potentially influence policy with the European Commission as well, and potentially more in the EU, and Shala and colleagues in EFAB with us at Roslin are really, at the moment putting together the reports from that. So there's opportunity to join that think tank if anyone really feels strongly about genome editing and has ideas to put to that.
And just one more question for you, Emily. Do you, from what you've seen here and what you know about how the AQUA-FAANG was translated in cattle, do you see what were the quick wins? Or I presume you were finished earlier than this project was?
No, so the BovReg project in AQUA-FAANG is focusing on cattle. I would say it finishes later, so it finishes in February. Okay. But the big wins have been the Australian groups who had the 1,000 Bulls Consortium, which was initially a whole genome sequencing project and big community buy-in. And having... So one of the easy things to do quickly is to have genotyped populations that also have functional annotation information, so gene expression at scale. And then that gives you huge potential to do things like transcription-wide association studies to get to the genes under the under the QTLs or the EQTLs, a higher resolution. I think there's generally just a large community working on cattle doing those things. I think from in terms of I don't know if there's ever easy wins, unfortunately.
But that was an end of the AQUA-FAANG for cattle, was this 1,000 bull array, or whatever you want to call it.
So the array that Sig mentioned was actually developed for Holstein Friesian cattle based on functionally enriched information, and then had good applicability across populations or breeds, I guess. And that was developed from investment in Australia and through the 1,000 Bulls, where the whole global community added sequence information from many different populations to test the suitability of those SNPs. So it was really a community-led initiative, I guess. But it's separate from the cattle components of AQUA-FAANG.
Okay, yeah. Okay. I wonder, could we do something similar in salmon? No?
Just to complicate a bit, so...
it's not complicated enough.
I think so also what we see in farm animals like livestock is also the pan genome. So
the what? Sorry?
The pan genome. So what people are
pendulum,
doing...
okay.
Pan genome, yeah. So what people are doing is they sequence many individuals, whole genome, end-to-end, so telomere to telomere. And with the new sequencing technology, this is very possible. And what you get from that is a lot of structural variants that you can't genotype them on your array, so you need new genotyping technology to do them. But there's a number of species now where we see that the structural variants, that they are not necessarily in LD video SNPs. So sometimes you can't even target them with SNP in perfect LD. And there is a paper in Tomato saying that they explain a significant proportion of the heritability, so the missing heritability. And that's also what they are working with now in humans.
Okay, so you're never going to get that on your SNP array. You're never going to be able to capture that.
I think you can capture some of it, but not all of it. So you need additional genotyping technologies, I think. But this is developing fast. So, but I think the fact that we are not working with complete genomes, and this has been mentioned. I think Antti, you also talked about this. It will change in the future because the technology is now there to characterize the genomes in a much better way. So you get this information, but you need a way to deal with it in a proper way. And so the annotated pan-genome, that's what we need in the future.
Yeah, that's all. Any other questions, comments?
I have a very small, modest proposal about the last question, how to integrate functional genomics into our genomic prediction models. I think we could find a way to collaborate because in that respect, species doesn't matter. I mean, and it's such a big space to explore of solutions that maybe Tim was looking in one direction, we are looking in another direction, Andrea in another one. So I wonder whether we can sort of create like an informal community across species, because in fact, understanding which are the best weights or the best, say, functional notation to integrate should be, to a certain extent, be transferred from one species to another one. So I think we should try to keep in touch, and I agree with Antti that we need also computer scientists to improve the speed and the efficiency of softwares. In fact, we are collaborating now with the computer science department in Padova to develop some of those methods, because I mean it's not our job to improve the efficiency of the softwares. So we need to put there the genetic knowledge and biological knowledge. So this is just a small proposal, but I think it has been working across the three AQUA-FAANG project, GeneSwitch, BovReg, and AQUA-FAANG. We actually were really mostly benefiting from that collaboration. We had a dedicated work package. We had just Sarah testing some methods, but that could be something that might require some financial support, but not that many, and could be really cross-species useful.
I think it would require some kind of a structure or financial support, though, like a project like this, because everybody is completely stretched working in bioinformatics in the industry, just getting data out.
I have I have one which I can do whilst I walk. One thing we haven't touched upon, which might be interesting to get the perspectives of the industry people in the room, is in a hypothetical AQUA-FAANG 2.0 project, what could and should the role for industry be in that project? So maybe Antti and Emily could comment on how it's compared with AQUA-Impact and the AQUA-FAANG projects. But in AQUA-FAANG 1.0, we had 10 industry partners, and all of them, bar 2, including Select, had quite a small amount of funding, which basically covered costs to come to meetings like this. So it would be good to know what the industry people think their role could be, what they would like it to be, if at all, in an AQUA-FAANG 2.0 project.
Okay, if I comment on the behalf of the AQUA-Impact, then AQUA-Impact is an innovation project, meaning that we make limited new research, but we put the current knowledge into practice, typically within the breeding programs. And the way we constructed AQUA-Impact was that 40% of the budget went to the companies and 60% to the research institutes. And the way let's say the content was created was that we discussed with the companies that what are your needs, what do you want to develop, what is important for you, and then we discussed that if it's possible to include this activity into the project, and can the, let's say, the local or any researcher help there. So we created these pairs or triples or of partners who could actually test something within the company or using the company data or collect data with the company. And then we were actually able to kind of respond to the needs of the industry much better. And I think this sort of practice is very good. And if you are, let's say, a university researcher, then actually this allowed for researchers to get very unique data sets, because the company could provide something that they would never ever get elsewhere. So it was really a win-win.
Yeah, that project was definitely a win-win for the industry. I mean, it actually changed some of our practices from that. It was a very applied project. Quite different to this, but definitely was very worthwhile being part of. And just to your question, Joe, one thing we can provide that we're not utilising enough in industry, I think I've said this a few times now, is like the data in terms of the actual fish that we're collecting, either that are undergoing disease challenges in the sea. So like, for example, gill data. We don't run challenge tests for gill disease. We actually just observe thousands of fish in a trial unit, but the fish aren't killed, the fish are kept. So like what's happening with the heart? We don't have the time to do anything except look at the cells in the heart. That's all we can do. So there's a lot of phenotypes being generated, complex phenotypes and tissues that are not being utilized.
Yeah, my question was something that picked up from Ian's comment about economic analysis, because we know that the single most expensive component to farmed fish is feeding. And I think the selection for growth has been shown to improve feed efficiency, but there's a ceiling to which that occurs. So I'm wondering whether we're obviously missing something around feed efficiency. And so are any of the companies using truly measures of feed efficiency to improve growth performance and improve cost of production? And the second part is whether you can do something more advanced and actually be connecting the diet itself, so making a really low protein diet or made with alternative raw materials that can integrate genomics and selection with a low cost, low production feed around that concept, that just to be able to produce more with less inputs.
There was quite a bit of that in the Aqua Impact project. We had feed efficiency trials using magnetic beads. There was that Nutec Aqua, but it was, yeah. And we also had a number of projects associated with genotype and novel feed ingredients.
And Mark van der Poot in the INRA Labs has done a lot of work on feed efficiency, particularly in bass and bream. And it's very difficult to measure. Of course, you have to measure growth and feed input and excretion. And you need to have animals individually housed, which some animals don't like to be individually housed, and a huge amount of labour and cost. So he's been looking at various proxies like fasting tolerance and selecting for that, and showing it correlates very highly with improved reducing the feed efficiency FCR. And we've tried that across a number of bass populations and allied species, and it works quite well. It doesn't work in bream, but it does work in bass. So there are, to answer the question, there is quite a bit of work going on.
But definitely we could do a lot more there, because...
Yeah, so I know it's being, you can theoretically do it and it can be expensive. I think Benchmark had developed some stable isotope techniques that seemed a lot more amenable. I'm wondering whether they're actually being applied in industry currently.
Just one more point. There's a very interesting article, you could Google it, and it was produced by World Wildlife Fund, and it was looking at Vandermeyen tiger shrimps across Southeast Asia. And it was a very thorough economic analysis, biological and economic analysis. And if you could reduce the FCR by 0.1, it calculated how much land use you would save, how much water you would save, how much labour you would save, and for a couple of countries, how much more profitability you'd get through reducing the costs. It's a really well written and illustrated paper, so it's worth a read.
If I can comment on this one. To improve feed conversion ratio, you can think that there are like two lines of research. And one is that you get individual records of feed intake from the fish. Then you actually are able to measure the feed conversion ratio. There are ways to do this. It's challenging. The other line of research is that you can indirectly improve FCR. And this is what happens, for example, also in pigs, that if you select for rapid growth, you control the body composition, especially the fat deposition, and you try to improve fillet percentage, which means the protein growth. Then you are indirectly making the animal to use the feed more efficiently, and then you improve feed efficiency. And because, for example, lipid deposition and body composition growth are much more easy to record than feed intake, then this indirect way might be, well, it's already applied. I don't know if it's 100% well documented that it works as well as we might think. And if there are species differences, that perhaps it works better with salmonids, which are more fatty fish. But these two lines of research are used.
Okay, so I think that's us. So thank you everyone for sitting here at our discussion. And once again, thank you to the AQUA-FAANG partners for delivering this baby to us. Let's see how we rear it. We'll try not to mess it up too much.